We started out chapter 26 learning a little bit about the broad characteristics of animals. We talked about how they move a little bit. We talked about some of the big categories. We talked about some of our body plans that we see in our animal groups. The next thing that we're going to talk about are germ layers and development. During the process of development of an organism, they are going to produce a series of layers of cells called germ layers. These germ layers are going to develop into specialized tissues within the organism. Our animals are going to usually have 2 or 3 of these layers. We'll talk about what those layers are in just a few minutes. But these layers come about after fertilization of an egg by a sperm, where we produce what we call a zygote. Zygote is just a fancy word for a fertilized egg. And that zygote immediately undergoes some successive cell division. It's a very quick cell division called cleavage, and this cleavage forms a structure called a blastula. In our lab for animals, we look at a starfish blastula, and you can see that the shape is roughly similar to this diagram we have down on the bottom. Our cleavage goes from the fertilized zygote to several cell divisions resulting in the 8-cell stage. These 8 cells all have different fates in the organism. So each of these 8 cells are going to develop into a different part of the developing organism. Depending on how complex, it may be a lot of development, less complex, might have less development, less differentiation. But after that 8-cell stage, the organism becomes a blastula. A blastula is essentially a hollow ball of cells. And inside that blastula, we're going to form a structure called a gastrula. The gastrula is basically a pocket or an indentation that forms inside that bubble. So the process of gastrulation is an invagination of that outer layer forming the gastrula. And in this image here, we have that outer layer. That is going to be one of our cell types. We have an inner layer that is going to be a second cell type. And then in between, we have a potential third layer. So all of our animals are going to have at least 2 layers, the outer layer and the inner layer. Some of them are going to have this sort of middle layer. The inner layer of cells in that gastrula is called the endoderm. If we sort of make a quick sketch of our blastula here, we had that inner layer of yellow cells. This inner layer is called the endoderm. And then is this layer here. That endoderm is going to develop into the digestive tract of our organism. The outer layer, the ectoderm, is going to develop into the outer layer of the organism. So this is going to be the surface layer. We would think of this as the skin layer or the epidermis. It really depends on the organism. So if its surface layer is an exoskeleton, it might be the exoskeleton, but it is also going to give rise to the nervous system. So this includes brain and all of our parts of our nervous system that extend out from the brain. Inside the ectoderm, but outside of the endoderm, we have a layer of cells called the mesoderm. And the mesoderm can take a couple of different forms. It can line the ectoderm side, or it can line both the ectoderm and endoderm side of the blastula. The mesoderm is going to form all of the organs that are in between the ectoderm and the mesoderm. So endo means inside, ecto means outside, meso means middle. So your mesoderm cells are going to develop into all of the internal things that are not your ectoderm and, epidermis and nervous system, and also not your digestive tract. So this is going to be almost all of the organs and your muscles that develop out of this mesoderm layer. In between the layers, we also have a cavity. And the cavity that forms is going to be a little bit different depending on the organism as well. So some of our organisms have endoderm and ectoderm. Others also have the mesoderm. And then this cavity, inside this cavity, may be fluid filled or not, and may be completely lined with mesoderm or not. So I would like you to take a second, summarize these 3 different germ layers, see if you can quiz yourself, describe where they are in the blastula and what they are going to develop into later. So this is an image of an earthworm. We've got a cartoon here drawing of that earthworm. We've got a cartoon drawing of its insides, and then we've got a microscope slide that shows the analogous structures. So our cnidarians are our cnidarians have both the ectoderm and the endoderm. They don't have the mesoderm layer, which means that they don't have true muscles. They can't have true muscles because the mesoderm develops into the muscle cells. So we call cnidarians diploblastic, meaning they have only 2 layers to their blastula. And all of our bilaterians are going to be triploblastic, meaning they have all 3, ectoderm, endoderm, and mesoderm. So our earthworm is a really great example of all of these layers. It's relatively simple. On the outside, this blue layer is going to be the ectoderm. We can see that blue layer here. It's not blue in our image, but this is where the blue layer is on the outside. So it surrounds the outside of the organism. Inside, we have that digestive tract. That is going to be our endoderm. It's a little harder to see the layers here, but you've got a layer of endoderm here. And then inside this area here, you have the digestive tract. So that endoderm is going to surround the digestive tract. Surrounding that digestive tract is the mesoderm. The earthworm has mesoderm on both sides, surrounding the digestive tract, and then also lining the ectoderm. So we see it in 2 locations. The body cavity that I mentioned is this area between the 2 layers of mesoderm, between the endoderm and the ectoderm. In this particular organism, we say that they have a coelom. We'll get this in our notes in just a second, but a coelom is lined on both sides. So lined on the ectoderm and lined around the endoderm. So coelomates are organisms that have a true coelom. A true coelom is completely lined on both sides of their cavity. So we've got that diagram of our worm again. We've got the ectoderm on the outside, endoderm in the center, and then you can see the red mesoderm lines the inner layer of the ectoderm and the outer layer of that endoderm. So if we have lining on both sides of that cavity, then we say that this is a true coelom, and the organism is called a coelomate. There are two other options that are not lined on both sides. A pseudocoelomate has lining only on the ectoderm side. So we only see that mesoderm lining the ectoderm. It does not line the digestive tract or the endoderm. Pseudocoelomates have fluid inside their body. This is a fluid that is going to help fill up the space in the organism. We see that fluid in the coelomate as well. So this is kind of what gives these worms their structure. They don't have bones, they don't have exoskeletons. This fluid is basically a non-compressible fluid that helps to give them structure. Lastly, we have acoelomates. Acoelomates are organisms that have the mesoderm lining the ectoderm, but they don't have a fluid-filled body cavity. So both our pseudocoelomates and acoelomates lack that second lining. The difference between them is whether or not they are fluid-filled. So our coelomates and pseudocoelomates are fluid-filled. Acoelomates have no fluid. So let's go back to that gastrula for just a few minutes. On the outside, we have the ectoderm. On the inside, we have the endoderm. The endoderm is essentially a cavity. There's nothing in it in early development. Remember that this blastula is a hollow ball. And then when we form the gastrula, we basically form a hollow ball inside a hollow ball. But there's this little opening right here that is formed through invagination. And this endoderm that we have is going to develop into a digestive tract. So eventually these cells are going to kind of grow to the other side, and we're going to get a tube that runs down the center of this organism. That tube is going to be a tube all the way from the mouth to the anus. So your digestive tract starts at your mouth, your waste comes out at the other end. That blastopore that we form can go one of two directions. That first indentation can either be the mouth or it can be the anus. There is not really any reason for one over the other. It's just how the development is happening. There's not an advantage to form one first, at least not that I know of. But our next category of organisms that we're going to talk about are categorized by whether the mouth or the anus forms first. So our protostomes, proto means first or before, stome means mouth. So in our protostomes, the blastopore becomes the mouth. In other words, the mouth forms first. In our deuterostomes, deutero or duo means second. So this one means that the mouth forms second. If the mouth forms second, then this indentation has to be the other end, which is the anus. So in the deuterostomes, the anus forms first. And when that blastopore extends through to the other side, then we get the mouth. On this one, we get the anus on the other side. So both of them are going to develop a digestive tube that goes all the way through the organism. Just depends which one develops first or what the gastrula develops into. Finally, many of our animals are segmented. Segmentation means that the body is divided into specialized regions. Now, these regions might be similar, they might be a little bit different. And the reason for segmentation is that it allows for specialized regions. In other words, we have regions that can do specific jobs within the organism. If you think back to that cartoon image that I showed of the earthworm back on the coelomate images, that is an organism that is segmented. All of the segments are the same. Whereas if you think of many insects, we see segmentation very frequently in insects. Insects have a very distinct head, a central part we call the thorax, and then a back end we call the abdomen. When we have this specialization, this allows for differentiation of structures. And very often we have segments that are dedicated to complex movement. So this allows for some more specialized structures for locomotion. So we start to see appendages, we start to see segmented legs, we start to see things like antennas. We also might have repeated organs in some of our segments depending on the organism. This is going to be useful in case a region gets hurt, damaged, something like that. So we'll talk about segmentation a little bit later in more detail when we get to our arthropods, which are our insects. Our segmented regions are usually going to be separated by something called a septum. A septum is a structure that separates cavities. So for example, you have a septum down the center of your nose. It separates your nose into two different nostrils, one on each side. Septums are usually a little bit flexible, a little bit soft, like maybe cartilage or something like that, that is going to separate the different segments.